I reckon we can probably make a start because the first few slides are intro slides anyway, and then we can slow down if need be as we go. So let us start. So today we are having a look at one of the Micron's papers. This one, Functional Connectomics Reveals General Wiring Rule in Mouse Visual Cortex, which I should admit, despite being a neuroscientist, I found pretty hard to read. I guess my background is in behavioral neuroscience and cognitive science, and this is very much a hardcore sort of connectomics paper. Some of the details about the mice behavioral aspect made sense to me, but otherwise I really had to dive into it, which was good. It caused me to learn a lot about how these different practices and procedures work. But I might need a little bit more help from the audience today if I've missed something or if I'm not saying something that's quite right. So don't assume I know what I'm talking about just because I'm the one who's presenting. If something sounds wrong, speak up. Before we get into the details of the paper, just again, this is one of the last journal clubs we're going to give this year before we have our meetup for the year. So as a reminder, we're having these journal clubs because we're interested in this memory decoding challenge. We want to see the first demonstration of decoding a non-trivial memory from only a static map of synaptic connectivity from a static brain. Now, it's very unlikely anyone's going to win that prize in the few weeks that exist between now and when we have our meetup for the year. But we also give away these annual research awards, 4 of them, to honor and highlight research into how learning and memory are physically encoded in the brain. Basically, people who are making these milestone steps towards being able to do this decoding from a static brain. And we are going to have an award ceremony in San Diego at the Society for Neuroscience event on Monday, the 17th of November, and the details are there. And if you are interested in this presentation, then you are very much going to be welcome and interested in the meetup. We'll have a panel, there'll be a discussion, there'll be a bit of socializing, there'll be some prizes, a very brief prize ceremony. It'll be fun and interesting. So, before I get into the paper, it's worth making clear that this paper we're talking about today is part of a set of papers that were all released in April of this year as part of the Microns project. And this Microns project was an attempt to try and map the full connectome of about a cubic millimeter of mouse brain from the visual cortex, something like 200,000 cells and 500 million synapses in this region of the mouse. But what's also interesting is, unlike other sort of connectome projects, like for example the Flywire, so the full map of a fruit fly connectome that was published last year, they also had a whole bunch of functional imaging that came from this same mouse that was then imaged structurally. And that's pretty critical for the paper we're going to talk about today. So they had things like long calcium recordings from the same, or at least a subset of the same region of the visual cortex before they went on to do structural imaging. And that means that across a series of papers, they were able to look at all sorts of connectomic information, but also functional imaging, functional information. And so, for example, they have papers on analyzing sort of just the structure of this cubic millimeter of mouse brain, whether that's in terms of the kinds of neurons that are present there or what they could find from mapping out the regions. They had this association between structure and function papers, which is what we're going to talk about today. The top one being the key one we're looking at, which is trying to look at, can they determine anything about why particular neurons have the particular responsivity that they do based on their structure and their connections to other neurons. Critically related to the paper we're talking about is they also built this foundation model, the same related things to these large language models. They built an equivalent sort of model for predicting the neural activity of these animals. And we'll get into that a little bit more as we go. They also have a bunch of improvements in, for example, just doing connectomics at scale. So they have this cool new sort of Git-like versioning system for actually keeping track of all the annotations that were happening during the manual proofreading, of trying to clean up the data that they received, or doing automatic tracing of the neurons. And they have a bunch of other tools, which is to say, this is an enormous amount of work. All of these papers have like 30 authors on them, some of which overlap between each of the papers, but it's a huge amount of effort went into this particular project. But anyway, we are focusing on this specific paper, looking at functional connectomics revealing general wiring rules in the mouse visual cortex, by Zhuokun Ding as the first author. And on here, there's people from Baylor College in Texas. There's people from the Allen Institute. There's people from Princeton. There's people from all over the place. A lot of neuroscientists went into doing this. So let's start. So I mentioned that this paper involves both functional data and structural data. So to set the picture initially, basically what they did is while the mouse was still biologically active, they did repeated imaging of an area of the visual cortex. So this area V1 here, and then adjacent higher visual areas of RL, AL, and LM over sessions where they showed the mouse repeated video clips, short video clips, either of sort of natural movies or of like artificial parametric stimuli, think things like Gabor patches. And then they measured thousands and thousands of thousands of different single units. They mentioned that their imaging technique could probably go down to large dendrites, but in this circumstance, they're just looking at sort of single neurons. They perform functional imaging on these neurons during these sessions. They also keep track of some of the anatomical markers of what they're looking at, things like blood vessels, so that later on, when they go and do electron microscopy on a subset of this brain region, they can also stitch the two together and line them up and know where which cells correspond to each other from the two different data sets. So you can see here, the green box in the center corresponds to where they were able to do functional imaging, and the gray box corresponds to where they've done EM structural imaging. So there's not a complete overlap in the two sets of data. It's a subset of the data which corresponds to where they're able to map the two onto each other. But it's that subset that we're going to be looking at throughout the course of this paper. And specifically, what this enables them to do and what they were interested in doing throughout this paper is asking a few different questions about how the activity of presynaptic neurons is related to different either postsynaptic or unrelated control neurons, as they put it. So imagine, for example, you have a presynaptic neuron, this neuron here in yellow, and it projects into a certain area, and it can do a few different things. It can make a synapse at some point with a postsynaptic target, which is labeled here in black, and the little connection is here in this circle. It can feel like it's almost made a synapse with a different neuron in that their axon and dendrites travel together, like they become very close to each other, but it might not actually connect to that other neuron for whatever reason, whether that's because it just never happened to structurally connect, or it once had a synapse, but they've degenerated since. For whatever reason, you have sometimes these axon dendrite co-travel control cells. So these cells where it's like, they could have been a synapse, but there wasn't. So that's what they're labeling here in red. And specifically what they quantify for all these cells is the axon dendrite co-travel distance, which is basically like how long or how frequently did the axon of a presynaptic neuron travel in sort of tandem with the dendrite of a postsynaptic target. And they do that both for cells that they actually did connect to, so the actual targets, and for cells that they didn't end up connecting to. They're just quantifying that in general. And then they also have these same region control cells. So these are cells that are in the same, they're in part of visual cortex, but which the axon of this presynaptic neuron never really gets that close to. So it doesn't count as being a axon dendrite co-travel control. I think their threshold was 5 micrometers from memory for what they counted as whether it had a significant co-travel distance or not. And you can see an example here, a sort of structural example of what that actually looks like when pulled out of the electron microscopy data. To give you some sense of what these different examples look like, you can see labeled in the yellow triangle is a particular presynaptic neuron, either in V1 or in HVA, which means higher visual area. So that's any of the RL, LM, or the other regions they were mentioning before. And you can see sort of the axon, which is labeled in gold, of this cell projecting out both to within the same region, if it's V1, or within the same region in HVA. And you can see all the different targets it makes and whether it connects or not. So the colors are the same. So the presynaptic neuron is in gold. The postsynaptic targets are in gray, and the ADP controls are in red, and the same region controls are in blue. And you can essentially just see that there's a huge number of examples for each of those 3 different types inside any brain region for any given cell. So they have plenty of data to work with for their analysis. I will just check everyone is still with me before I continue on into the results section. I'm seeing a thumbs up, so that is good. Okay. Now, one of the critical things, and this has got me a little bit confused when I initially started reading the paper, is that a lot of the comparisons that they are making in terms of functional data in the paper is not back to their actual calcium imaging data that they took from the mice. It's a comparison to this digital twin model of what they think the neurons will do when exposed to particular sort of video signal. So basically, to make this clear, what they did is they trained, they took from this mouse and other mice a whole bunch of calcium imaging from the mouse as it was watching these various different videos. And at the same time, they also tracked the mouse's movement, and they also tracked where its pupil was looking at and what the size of its pupil was. And they fed all of this data into a foundation model, which attempted to predict the calcium responses of the neurons that they were tracking during the imaging session. So you can see, for example, up here, up top is an example of a neural trace where the green line is the actual in vivo calcium fluorescence data that they might see for any particular neuron that they're able to trace. And in blue is the sort of in silico guess of this model as to what the calcium is going to look like, based on being fed in all this information about the animal's eye position, what its guesses to the perspective of the mouse is, through these various different sections of first trying to display the perspective of the animal, then do a whole bunch of post-processing. Yes, Randall?
Yeah, I don't know if you needed this input because it's just that you mentioned earlier that you were a little bit not sure why they were doing this. But by building up this model that produces the same kind of response for the functional imaging that they did record, they were then able to also run these additional sets of rich stimuli and videos through that network that they created, the recurrent network, to get more indicators of what they should expect. So it was basically a way to enrich the number of functional experiments they could carry out.
Yeah, they make that quite clear. They do in all sort of the extended data figures. They show a comparison where they use the actual calcium imaging data from the mice for a lot of things to compare it to the in silico data to sort of validate that what they're seeing is not just a function of them having built this foundation model. But I guess what I wanted to make clear for people in particular is that a lot of the comparisons that I'm going to be talking about when they talk about the functional similarity or the similarity of the responses of these neurons is inferred from what the model says that the response pattern or the feature selection of that particular neuron is, rather than directly from having observed the calcium imaging for that particular neuron, which is just one of the things that tripped me up initially when I was reading the paper. So I wanted to flag that for people. But thanks, Randall, for jumping in. Yeah, so foundation model is predicting the activity of this neuron, in particular when shown sort of arbitrary videos. So to Randall's point, they were only able to show the mice like a limited subset of videos, but they're going to use this model to predict that neuron's activity in response to all sorts of different videos. And then they can compare what that neuron does for one video to another in comparison to a different neuron somewhere else in the visual cortex. And that's detailed mostly in this other paper in the set. This, I don't know if you can see my mouse moving. Yeah, you can. It's a foundation model paper. So to get into the start of their actual data, what they're showing here on the X and Y axes is the degree of signal correlation on the X axis, which is basically how correlated was the activity of two different neurons, as determined by that, in silico calcium fluorescence, so the responsiveness on the foundation model. And then on the y-axis is how much do these two different neurons have co-travel distance together. So how much does like one axon travel with the dendrite of another neuron in that region? And what they're asking here is if there's more travel distance between a presynaptic neuron and a postsynaptic neuron, are their responses more correlated with each other to a given visual stimuli? And the answer that you can see here is pretty clearly yes for all the different brain regions that they're looking at. But then they're asking the same question with not just like co-travel distance, but actually looking down at the synaptic level. Because theoretically, you could have circumstances where these axons and dendrites are traveling together for a period of time, but where, synaptically, maybe they're not connecting a lot, or maybe they're connecting to a certain degree, but some have higher synaptic density than others, and that might affect the responses that you're seeing. But what they're showing here as well is for all these different regions, that as synaptic density increases, you see an increase in the correlated activity between these different neurons. So again, that's not very surprising, but it will tie into what we're going to talk about in the next couple of slides. And just to give you a few examples of the other ways in which they end up quantifying this, they also look at things like the size of the synapse that they're looking at, whether it has a small cleft volume or a large cleft volume. And they see that the signal correlation, or the residual signal correlation, so the amount of signal correlation that's not explained by these previous factors, also increases as the size of the synapses increase. And they also see the same thing for the number of synapses that are made between two different neurons. So all this is to say so far that the more connected these neurons are together, the more similar their signal correlation is, which perhaps is not very surprising, but is good to see quantified down at this connectome specific level. So then we get into some stuff which is a little bit more complicated. So let's talk through these particular figures. So at the top here, we're again looking at axon scale co-travel distance. So how much there was a co-travel between a presynaptic neuron's axon and a postsynaptic neuron's dendrite. But this time, what we're looking at in the blue and in the gold is looking at the distance between the receptive fields of the two different neurons. So how far away were their receptive fields for visual stimuli centered? Again, according to this digital twin in silico model of where those neurons are looking at on the videos that are being presented. And And in blue, we're looking at the feature weight similarity. So what sort of features of the visual stimuli are these different neurons looking for? Are they the same features? Are they quite different features? And so from left to right for both of these, we're looking at less similar to more similar. And from top to bottom, we're looking at less co-travel distance to more co-travel distance. And what you can see is that for an axonal scale sort of data, so for when we're just looking at to what degree a presynaptic neuron's axon and a postsynaptic neuron's dendrites travel together, as that co-travel distance goes up in each of these different visual areas, so from V1 to V1, from HVA to HVA, or any of these, you can see that as they travel together more, both the receptive field distance decreases. I'm pretty sure that's how I'm supposed to interpret this. And the feature weight similarity increases. So the neurons are responsive to the same sorts of features, and they're likely to have receptive fields that are closer together. In contrast, that same relationship does not necessarily hold for down at the synaptic density scale. So in this circumstance, what they are saying is that factoring out the co-travel distance, if we just look at the density of synapses on a given region of the axons and dendrites, then how does that affect this sort of correlations? Does increased synaptic density also necessarily mean that the receptive fields are closer together and the similarity of features that these neurons are looking for is increased, or is that not necessarily the case? And what they find is that it still holds for the feature weight selectivity. So increased synaptic density still implies increased sort of selection for the same visual features, but it doesn't necessarily hold the same way for this receptive field distance. So it seems that the synaptic density doesn't say anything about whether the positions of these receptive fields. And it's interesting that these come apart, this axonal scale information and this synaptic density information, sort of implying that at the bigger scale, you're seeing these things which imply things like retinotopic maps in the visual cortex, but down at this synaptic density level, that sort of goes away when you get to the real nitty-gritty specifics of what the neurons are doing. And then from memory, this is, so this is quantifying as either the synapse size gets bigger or as the number of synapses get bigger. And they're showing the same sort of thing here, that what an increased number of synapses or increased synapse size seems to be implying is more about what sort of features of the stimulus the neuron's looking for, not anything about the receptive field positions specifically. So these plots are quite complicated. So again, I'm just going to check everyone's still with me before I move on to the next slide. Yeah, go for it, Ken. You're muted. All right. No, you're not.
This is fantastic. I want to make sure that I understand in B. So in A, the y-axis is the amount of distance that this axon co-travels within 5 microns of the target dendrite.
Yeah.
And in B, this is synaptic density, so number of synapses per co-travel length. It's not per.
That is my. Yeah, I think that's correct. So they specify that because there is quite a difference in this co-travel distance between various different neurons. What they look at is they look at sort of the. I'm going to have to try and remember the method specifically. Yes, I think they're controlling for the level of co-travel distance. They're then looking at the average synaptic density given a unit of co-travel distance. And then they're looking at as you decrease or you increase the synaptic density compared to the average, how do these features change? So that's why it's centered on 0 on the x-axis, and we're just looking at for a given unit of co-travel distance, if the density is higher or lower, how is that affecting these features? So yeah, I think how you described it is correct.
Okay, and another way of just you said this, but I want to highlight this. Subset A there, figure A, is really kind of stuff that is built in. You can imagine by development, and B is potentially heavy in plasticity, quote unquote heavy in plasticity, and the fact that that there is a negative correlation between receptive field size or receptive field position is kind of saying, this is a question, let me phrase it in the form of a question. It's kind of saying that the brain doesn't want to have two features that are exactly the same, that are looking at the same part of the visual field, because that's not efficient. And so they're actually pushing those things away, maybe like a self-organizing map type of algorithm, where is that a way of interpreting this?
I mean, I definitely think that is potentially one. I definitely agree that the authors make the same claim about the retinotopy initial guidance from development might be different to what they're doing down at the fine synaptic density scale. Precisely whether it's about efficiency, I mean, that makes sense to me, but I am not an expert in this. But maybe Randall has some thoughts. Go for it.
No,
Randall.
not anymore. You already said what I was going to say.
Cool.
This is just one more thing. The analysis blows my mind on this paper, because the idea that they have the ability to track the amount of distance that all the axons are within 5 microns of the dendrites is just absolutely crucial to this analysis. And they would get a completely different answer to this, as you see, if they didn't have that analysis. It's an amazing paper.
Yeah, I should also make clear for everyone, because I didn't specify this at the start, that this analysis is done only on a subset of the neurons for which they had both the functional imaging and structural imaging in this region, and that they were able to trace to a reasonable degree. So it ends up being presynaptically, I think it's only a few hundred neurons or something, and then they look at all the postsynaptic connections. So it's certainly a large number of neurons. It's not a trivial data set by any means, but it's not equivalent to they have literally got every single piece of data out of this region. And it would be interesting to see once they hopefully do have some sort of completion of mapping this. I don't think the results will change, but it might just tell them even more that they already have. I saw a hand come up.
I'm sorry, that was me. I pressed the wrong button.
Okay, cool. All right, well, let's continue then. So this is sort of a quantification of, again, what Ken was just talking about, these differences between what they see at the axonal scale and at the synaptic scale for a whole bunch of different things. So again, these plot is a little overwhelming to look at initially, so let's just break it down a bit. So these two columns are looking at things at the axonal scale versus looking at things at the synaptic scale. And it's for different features. So it's signal correlation overall. It's the feature weight similarity of the neurons that are being examined. And it's the receptive field distance at the bottom, the distance between those receptive fields. And then what they're looking at the left on this axonal scale is the co-travel distance. So a smaller box means they travel together for less distance. A bigger box means they travel together for more distance. And on this right one here, it's about the synaptic density. So smaller box means lower synaptic density. Bigger box means higher synaptic density. And so what they're looking at is the like-to-like coefficient. So basically, how similar are the response patterns from less correlated to more correlated to each other, or less similar to more similar. And so basically, they're asking for a given brain region connecting presynaptically, connecting into a given brain region and layer postsynaptically, as you change the synapse density at the synaptic scale or the co-travel distance at the axonal scale, do responses become less similar or more similar? And they show that for signal correlation, it looks like the same pattern at the axonal scale and the synaptic scale. So let's say if we look at visual cortex presynaptically layers 2, 3, connecting to layer 5 of a higher visual area, you see this medium-sized co-travel distance and a medium level of like-to-like coefficient at the axonal scale, which is even larger at the synaptic scale. But in general, the patterns look quite similar at axonal, synaptic, for signal correlation and for feature weight. But where they diverge is for this receptive field center distance in the bottom here. And again, as Ken was saying before, this would be very hard to tell unless you had a proper dense connectomic map of what was going on here. Otherwise, you'd be limited to just, for example, this axonal scale 1 on the left, which would give you the right idea for signal correlation and feature weights, but not for receptive fields. And then sort of to get towards the end of the data, before we get into talking about the big picture implications of this sort of work, what they wanted to ask themselves is, given a particular presynaptic neuron and its postsynaptic partners, and all the different ways in which these neurons in the visual cortex can respond to visual stimuli, is it the case that postsynaptic neurons are more similar to each other than the, hold on, this one I have to make sure I describe properly as well. They had two different theories. So there was a presynaptic neuron that was projecting onto these postsynaptic neurons, and you can look at the activity of the presynaptic neuron and the postsynaptic neurons, and you can ask yourself, how similar are these postsynaptic neuron activities to each other, given they're coming from this presynaptic neuron? They could be very similar to each other. They could be less similar to each other than you would expect based on this presynaptic input that they're getting. And I've gotten myself a little bit confused about the exact question that they were asking again. So I'm going to try and muddle through it. But if anyone else who's read the paper feels like they're really on top of it, you can jump in now. But basically, they were looking at the connections, and they were looking at sort of a naive model of how functionally similar they would expect these sorts of neurons to be, and their naive model of their connection probability given these functional similarities. And they use that to, through, I think, some sort of like Poisson model, calculate their expected degree of similarity to each other. So basically from just like the degree of connection between them, they had some naive model of how, the degree of functional similarity between those neurons, which they compared to the in silico model they got from training the foundation model. And what they found was that actual neurons were more correlated, these actual postsynaptic neuron activities were more correlated with each other. So they had higher signal correlation, a little bit higher, than you would expect based on just the naive model of their connections to each other. I will admit that I got a little bit lost on this section as to the exact technical details of the implications here and how they did it. So I don't know if anyone else understood it better than I did. But the implication they said, and given I couldn't fully understand it, I'm just going to have to trust them, is that there is a higher degree of correlation between these postsynaptic neurons than you would expect naively, which means that there has to be some sort of more complicated rule for determining like their activity and or how they're connected to each other than, I think, very simple sort of, I'm not sure. I didn't really understand it.
Yeah, I can't say that I fully get it either, but this is, I think, where they then postulated that that kind of indicates that there's a higher order organization that's going on. So maybe this is where learning then combines into, I don't know, potentially that leads to more complex features or something. I'm not sure where that goes. Maybe Ken knows.
I don't have anything to add. I think you've covered it, or
All right. Well, we'll need to grab one of the authors at some point, get them to walk us through exactly what this meant, or spend a bit longer trying to really drill into the different aspects. I mean, I do feel a bit better when I see 30 different people on like an author list. I'm like, okay, it's not like any single person is like totally on top of all of this, maybe. But whichever of the 30 understood this the best, we can find them and get them to explain it to us. So that is an overview of the data and the methods they used, but I want to leave plenty of time for sort of discussion of the implications of this work and the micron stuff generally. So I'll just quickly go through sort of the implications. So the key findings are they find this like-to-like connectivity universally across the visually responsive neurons in V1 and these higher visual areas. They find that feature similarity rather than receptive field overlap is what's associated with this increased synaptic density down at the very small scale. And they claim that there's these higher order connectivity rules that exist beyond simple pairwise similarity that are determining these signal correlations. But that was the last bit that I just mentioned that I didn't fully understand. So some of the limitations are that a lot of this analysis is done using this digital twin reconstruction that was limited to the neurons that were functionally imaged. So they can't take this digital twin naively and start applying it to other neurons that are present in the visual cortex to know what they were doing. They'd have to retrain a model on those neurons if they somehow got functional imaging or data for them, or reconstructed their connections to the other neurons in a structural sense. Again, this is only looking at a subset of the neurons in this volume. And lastly, they flag that tracing the axons fully, getting rid of all the merge errors, is a very difficult job that required a lot of human proofreading and is incomplete. And there could be missing connections between neurons that aren't part of their data set. Now, some of that will be because the axons extend beyond the boundaries of the volume they were looking at. And some of that will be just because they've missed doing the full reconstruction yet. So some of these effects might be influenced by connections being absent that actually are present in the actual biological material. And that then, given those limitations and caveats, brings me to what Ken and Randall and I were discussing. So sorry to put Ken on the spot a little bit, but this is one idea of maybe how you could take something from what this sort of work was and apply it to the idea of doing memory, which I put in inverted commas, or at least like function decoding from the experimental data. I will say the more I thought about this, the more it actually reminds me of, and maybe this is my misunderstanding, but it reminds me of the sort of work we talked about right at the start of the year, the Lapalainen paper on looking at recovering functional activity from fly visual cortex structural images. So let me get into it, and then we can discuss whether I think that analogy, whether you think that analogy holds or doesn't hold. So imagine you repeat an experiment of this sort, but for a subset of the neurons in the visual cortex, you withhold the calcium imaging data. So you don't allow the information about the activities of those neurons, response to videos, to make it into your imaging data sets or your digital twin model. And then you do everything the same as the paper. So you map all the neuron connections, you create this digital twin to determine the functional properties for the non-withheld neurons. And then what you do is for your withheld neurons, you predict how they should be responding to visual stimuli based on their connection properties to the other neurons. So for the other neurons, you know where their receptive fields are supposed to be, you know what their feature similarities are, you have all this information, and you know how they connect to your withheld neurons, and you say, oh, okay, because it's connected to these neurons with these feature selectivity properties, and because it's connected to these neurons with these receptive fields, we know that actually there's a bit of a negative correlation between receptive field distances, and we know that there's this positive correlation between features. So I predict this withheld neuron will have a receptive field here, and it will have it'll be looking for these features. And then you validate and you score that. So you look at the actual results from the calcium imaging, and then you can do something like compare your actual results to a shuffled version of the data to give you a sense of like how good your score was, and maybe you can convert that to bits extracted somehow. Although in the email exchange we were having a little discussion about the how you might actually practically end up doing that. And yeah, the reason why I thought this was a clever idea that Ken came up with, or at least described, and it did remind me a lot, once I reflected on it, of what they were doing in the Lapinainen paper, which from memory, it's been a while since I looked at it, they had the entire visual cortex of the fly mapped out, but they didn't know all the various parameters for how the neurons related to each other. And they had some clever way, and Randall might remind me, or might remember better, of exploring the space and working out how individual neurons were supposed to actually be responding to visual stimuli that was largely constrained by the connectome information they did have. But I don't remember how they did their validation, and so like how they checked how they were doing. But yeah, this I think is a clever idea. It's different to doing, the most clearest example of memory extraction of like taking a mouse or taking a songbird and reading out the song that it is singing from its cortex, auditory cortex somehow. But it's still, even something like this would be a good demonstration of being able to look at the static map of a brain, look at the connectivity pattern, and be able to tell you something about the functional properties of those neurons in a way that would count as some degree of decoding from a static structure. And, if you did it really well, might be arguably worth winning at least some sort of prize. I will open for comments and questions here. So Randall, go for it.
Yeah, I just have two things to add. So first of all, what you were saying about Lapinainen, it's been a while, so the actual absolute details of the study, I'd have to look them up again. But so they had a bunch of similar structure in there. So they replicated that a few times because they found the average of that, basically a good representation of a piece of circuitry, multiplied that a few times to create a grid. And then they trained a decoder so that they could actually run experiments on this simulated silicon version of their reconstruction. And then indeed they were able to make some predictions. And those predictions, they were then able to check against, I don't remember if there were any new studies done or if it was just against prior studies that have already been in the literature. So that's something I'd have to look up. So that's one comment. And the other one is mainly aimed here at those of us, which is I think maybe half of the people who are here right now who are at Carbon Copies and who've been involved with the Brain Emulation Challenge. When you read through this here, you might notice that this looks almost exactly like what we're doing on our platform with the Brain Emulation Challenge, where we build a synthetic ground truth, something that is the equivalent of what they did here by reconstructing from that piece of tissue, right? And we hide a function within it. So we have the functional, this calcium imaging, right, that's going on in the middle somewhere. And then we tell researchers, go ahead, tell us what's going on in there, build this network, and then we can compare. Right, so there's a large degree of similarity between this memory decoding experiment and that validation of methods thing that we're building. So this, to me, this is beautiful because this seems like there is indeed a lot of overlap between the memory decoding challenge and the brain emulation challenge.
Yeah, one should always be careful in science with claiming precedence. And when I say Ken's memory decoding experiment, who's to say whether that's not just a rehashed version of what we've heard before, or my interpretation of what I've heard before? Sorry, Ken, go for it. You guys can just freely talk. You don't have to put your hands up.
Yeah, I would definitely agree. I mean, I'm essentially taking a bunch of discussions that I've had before on this journal club and really just trying to logically apply it. So for example, in a very general case, there's a memory decoding experiment is always going to have something to compare to, right? There's going to have to be some ground truth. So it makes sense that you hide some of the calcium imaging. Yeah. You don't want to hide half the calcium imaging data on the same neurons, because then you wouldn't be dependent upon the synaptic connectivity alone, right? So that's a key thing. If you compare, the one thing that I think people would have an issue with is using the calcium imaging at all, right? It feels like you're cheating, right? That you did a lot of calcium imaging on these neurons, and now you're trying to use that and the connectome to predict these other neuron responses. And I would argue that in the other experiments that we've talked about, it's a proxy for the same thing. So, for instance, we talked about using viral injection of the auditory cortex at some point to label cells that were responding to particular tones, and to use that in a way to decode the responses of lateral amygdala neurons. Well, that's kind of the same thing. If we had, it's substituting for some way we have to label some cells to get a foothold. And that might be that we label, maybe we don't have to label because there's some kind of tonotopy or retinotopy. If we think back to one of the papers that won last time, the Tony Zadar's lab that decoded whether the mouse would lick right or left, it was because they saw a tonotopy and a bias to looking right and looking left. They could see at a macroscopic layer in the level in the anatomy, and so you know, even if you got the full connectome of the animal, what you would be doing is you'd be going all the way out to the sensory surface, like the retina, and saying, okay, now we know what that function is. So I think we almost have to just accept that there has to be, for any decoding experiment, there's going to have to be a foothold that we have, and there's going to have to be some functional data that we are somehow hiding in order to compare. So, and the only other thing here is, I think I don't think they would have to retrain anything. I think they could literally do this analysis. The only real question is what half of the data, or what quarter of the data, what 90% of the data that they hide. I suspect that they would run into statistical problems where they don't have enough connectivity to really constrain stuff. But it would be very interesting to see how far they could get with something like this.
In reading the paper, Ariel, did you see whether or not the number of cells that they were able to use here, so the amount of presynaptic cells projecting to postsynaptic cells that they were actually able to use, if that was literally the maximum that they could do because of what was in the data, or if it was just that was the maximum they could do basically before publishing because of the amount of work involved? Because maybe you can still do more, and then the statistics would get better.
Yeah, I'd have to check again. They do make it. I would have to check.
Clear to me. I mean, they were definitely throwing out cells that didn't have good unambiguous calcium data, and they were supposedly throwing out a bunch of cells that didn't have good.
But it seemed like the final reduction when it went down to a much, much smaller set of something like 148 presynaptic, 2000 something postsynaptic, that was based on the difficulty in tracing the axons, not on the number of cells that were in that overlap area, if I recall correctly.
Yeah, I'd have to pull that up, but
so maybe there's even more information to be gleaned from just from the data that's already there, and then make a more statistically sound experimental suggestion. Yeah.
And maybe we should tell them they can win a memory decoding.
Yeah,
at
all
least
they have
one
to
of
do
the
is invest a
prizes.
few more $1 million, and then they can win the prize. Yeah,
they can win 25 grand or something, maybe even 100 grand. It's a good return on investment. Maybe another Nature paper at least. I wonder what a Nature paper is worth. Were there other aspects to this as well? Well, if we've got a few more minutes, the one thing I wanted to flag as well, which again, Ken was actually the first to point out, but I did notice while I was reading, is I enjoyed the end of the conclusion. They talk about how this data set is useful both generally going forwards, both the structural and the functional data, and also their immortalized digital twin that they created by doing this sort of foundation model production from the behavioral data and the calcium data. And it's a fun choice of words, this immortalized digital twin that, is in no way relevant to any of the interests of the people that are here about the implications of why they may have used that word choice. And that's all from the slides. So we've got 5 minutes left. We will have to finish pretty much on time today because I have another meeting to go to immediately after this one. But we can continue discussing. If anyone else who hasn't already spoken has any questions or thoughts, otherwise we can always finish up a little bit earlier.
You have to get somebody from that team on the panel discussion at SFA.
Yeah, that would be excellent. Yeah,
good. Let's have a look at who everyone is. So do you know the microns people, Ken? I don't know to what degree.
I know quite a few. I'm actually meeting a couple of them on this Friday.
Oh, cool.
I'll ask, but it's 30 people, right? So that doesn't say much. It's
yeah. I know I've emailed at least one of the people on here, and I know that Randall has been maybe emailing at least one of the people on here. I haven't received a response from them as of yet, but hopefully we'll get someone. We've got a couple of, actually this is for the other people here. So we got a response from Helene Schmidt so far, and from Andrew Payne are both confirmed as coming. So that should be fun. Bit of expansion microscopy and electron microscopy discussions. So yeah, should be good. All right, well, if no one else has any questions, then I guess we can just finish up slightly earlier. We may or may not have one more sort of journal club discussion before the end of the year meetup as well. We're still going to work that out. If anyone else wants to volunteer, as always, you're very welcome to do so. But otherwise, thank you very much for coming. Hope to see many of you there at the event at the end of the year.
Thank you for presenting, Ariel. I know it was a big paper to go through. You did a great job. Thank you.
Thanks very much, Randall. It's good for me to get my head into these things. All right. See you all soon. Bye.
Bye-bye.